Engineering Antibody Affinity and Cross-Reactivity by light chain shuffling. Welcome, Line.
Thank you, Chema. Do you see my screen?
Yes, perfectly.
Also?
Perfect.
Perfect.
Go on. Go on.
Well, I would like to start by thanking the organizers for having me here. I'm very pleased to have all of your attention to this project that I've conducted during my PhD. As Chema said, I'm Line Ledsgaard, and I'm from the Tropical Pharmacology Lab in Denmark, supervised by Andreas Lausten. And today I'll be presenting to you my work on a human monoclonal antibody that neutralizes the lethality of Nycalthia whole venom. So just briefly, even though Cecilia also mentioned it in an earlier talk today, I just want to say what we do and what our aim is in a tropical pharmacology lab. And that is that we envision a world where traditional antiserum, which is of non-human origin, is associated with severe adverse reactions, is expensive to manufacture, and has a low therapeutic content, can be replaced with what we call recombinant antivenoms. Recombinant antivenoms, which have 100% humane origin, are safer to use, can be produced cost effectively, and have a high therapeutic content. So this is the overall aim of our research group, that we want to enable this transition. And during my PhD, I've been trying to tackle a small part of this project, which is focusing on the venom of the monoclid cobra, or Nycalthia. So Nycalthia is a snake that's venomous, that is the cause of many of the severe envenomings in South and Southeast Asia. It's part of the elapid family, and it has a highly neurotoxic venom. The venom of Nycalthia is quite special. At least I have not seen a venom that looks like this before. More than 50% of the venom of Nycalthia consist of long-chained alpha neurotoxins. And then smaller parts of it consist of cytotoxins and phospholipases. But definitely the most medically relevant toxin in the venom of Nycalthia are these long-chained alpha neurotoxins. Also quite uniquely, 35% of the venom of Nycalthia consists of a single long-chained alpha neurotoxin called alpha coprotoxin. And this toxin is the one that I've been working with in my PhD, and the one that I'll be presenting work on now. So firstly, I would like you to just have a little bit of information about how these alpha neurotoxins actually work. So here we have a neuromuscular junction where we have a presynaptic neuron that releases a neurotransmitter called acetylcholine. Once this acetylcholine is released, it binds a receptor called the nicotinic acetylcholinic receptor. Upon binding, that opens up the receptor, and there's an influx of potassium and sodium channel ions. This influx results in a depolarization of the membrane, which in turn results in the firing of an action potential, and that's how we have muscle contraction. However, if you are envenomed by a snake that has a highly neurotoxic venom consisting of a lot of alpha neurotoxins, what these alpha neurotoxins do is that they act as antagonists of nicotinic acetylcholinic receptor. So once they've bound the receptors, they prevent acetylcholine from opening the channel, thereby preventing this influx of potassium and sodium channels, preventing depolarization of the membrane, preventing firing of an action potential, and thereby preventing muscle contraction. That's also why if you're envenomed by one of these snakes that have a high content of alpha neurotoxins, such as Nycalthia, you will be paralyzed and die from suffocation because you won't be able to use the muscles that you need for breathing. So what I've actually done, and what this project is actually about, is making antibodies against this very, very present toxin, alpha coprotoxin, in the venom of Nycalthia. So this project started a long time ago, back in 2015, where we initiated a selection or an antibody discovery program where we used an scFv-based human library to discover an antibody against alpha-copratoxin. One of these antibodies that we discovered is what I'll be referring to as the CO1 clone, and it had promising characteristics in all of the assays that we conducted, and it showed a very promising profile. So we actually decided to test whether it was able to neutralize the lethality induced by alpha-copratoxin in mice. So specifically what we did was that we injected mice with 2 LD50s of alpha-copratoxin, which had been pre-incubated with this antibody CO1 in a 1:2 toxin to antibody molar ratio. And the results look like this. So here you see survival of mice that were injected with only alpha-copratoxin. And as you can see, all mice died within the first hour after injection. And you see that the mice that had administered the CO1 clone had prolonged survival. So this was not a fully neutralizing antibody, but it did show great potential. So what I have done in my PhD is that I've taken this antibody and had the aim of improving it and making it a better antibody that could possibly even neutralize the lethal effect of alpha-copratoxin. There are many different ways that you can go about doing an affinity maturation project. And the one that I chose is what is called chain shuffling. So here on the right, we have an IgG, which probably most of you recognize. It consists of a heavy chain and a light chain. And this outer part out here, that's what we call the scFv and what is used in our discovery programs. So we have the heavy chain and the light chain. What you do in chain shuffling is that you keep one of these chains constant, in our case, the heavy chain, and then we pair it with a whole new naive repertoire of light chains. So why does this even make sense? Well, the initial discovery program to find this CO1 clone was done using what is called the Ioannis library, and it has a diversity of approximately 10 to the 10th clones. Within this library, there's a heavy chain diversity of approximately 10 to the 8th, and a light chain diversity of 10 to the 4th, 10 to the 5th. That means that if we were to have the full like a library with all possible combinations of light and heavy chains, we would need a library in the size order of 10 to the 12th, 10 to the 13th. And that was not the case. That means that this CO1 clone, there's a very big chance that it is not paired with the most optimal heavy light chain combination. Since the heavy chain in most cases is what drives affinity and specificity of antibodies, what I did in this project was that I took the heavy chain of the CO1 antibody, and then I paired it with this naive library of 10 to the 4th, 10 to the 5th, different new light chains. And then I did a whole new antibody discovery campaign where I did new selections, and I did new characterizations and sequencings and reformatting. So I'm not going to take you through all of this in the interest of time, but I'll just give you some numbers so you see how the process went. I started out by testing approximately 400 clones for their binding to alpha-copratoxin, but also to non-specific proteins. From there, I sequenced 62 clones, out of which 14 of them had unique light chain CDR3s. Out of these 14 clones, I reformatted 13 of these to the fully human IgG format and the Fab format. And then I did a lot of tests to see how they retained their binding patterns as compared to them in their scFv format. I also did a bit of biophysical testing, and I also tested how well they expressed in mammalian cell lines. After I had collected all of this information, I picked my top one candidate. And this top one candidate went through a lot of in vitro and in vivo testing since then, which is what I'll take you through now. So first I wanted to see, and this is a clone that I'll from now on call BO2. And first I wanted to see, well, now I've gone through this making new libraries, trying to do affinity maturation, doing a new selection campaign, but has it actually worked? Have I been able to isolate an antibody that has improved affinity as compared to the parent? So I used surface plasmon resonance to determine the affinity between these two antibodies and alpha-copratoxin. So here we have the parent, and you can see that it had an affinity that was already quite low at around 4 nanomolar. And then here we have the affinity matured clone, which actually went to sub-nanomolar affinity of about 500 picomolar. So this affinity maturation project resulted in an antibody that had approximately an 8-fold increase in affinity. But what is affinity? Like, I need to know whether affinity, this increase in affinity also means anything when it comes to the antibody's ability to block the interaction between the nicotinic acetylcholine receptor and alpha-coprotoxin. So what I did is that I did a blocking assay determining this. And as you can see here, I tested different concentrations of antibodies pre-incubated with toxins to see how the antibodies block the interaction between alpha-coprotoxin and a subunit of the nicotinic acetylcholine receptor. And you can see that they did both block this interaction fully at higher concentrations, but that the affinity matured clone had a lower IC50 value than that of the parent. And actually, it had improved approximately 4-fold. So this increased affinity did result in increased blocking. But did that also mean that this increased blocking would actually prevent alpha-coprotoxin from inhibiting these nicotinic acetylcholine receptors and inhibiting the influx of ions? To find out about that, I went to a company in Denmark that's called Sophia Bioscience. They specialize in running electrophysiological assays, and in particular, they have developed these 2 platforms for doing automated patch clamp. And I used this machine called the Q-patch to assess whether these 2 antibodies were able to prevent alpha-coprotoxin from inhibiting this iron transport across the membrane. And what I saw was that both of these antibodies were indeed able to neutralize the effects of alpha-coprotoxin and enable whole cells to once again have a normal current running across a membrane when acetylcholine was added to the cells. And once again, I actually also saw a difference in the ED50 value. So at this point, I had seen that I had made discovered a new antibody that had improved affinity, improved in vitro blocking, and now also I could see approximately 3-fold increased in vitro neutralization. But does this actually correlate with in vivo neutralization? At this point, I was quite pleased with these results and I was quite hopeful. So when we started planning the in vivo experiment, we were actually quite ambitious. Because Nycalthia, as I showed you earlier, has a not that complex venom, and the majority of the toxins in the venom are these long-chained alpha neurotoxins, I decided to, instead of testing whether this antibody would be able to prevent lethality induced by alpha-coprotoxin in a purified form, I wanted to test against whole venom. So what we did was that we incubated 2 LD50s of Nycalthia whole venom with different molar ratios of this antibody. Then we injected it intravenously and we monitored the mice to see their survival. As controls, we also injected venom alone, venom in combination with an irrelevant antibody, and venom in combination with an antivenom, commercially available antivenom. And now I'm very excited to show you the results of these experiments. First, I'll show you what happened within the first 4 hours. So the 2 groups receiving either venom only or this irrelevant control antibody, all mice died within the first 30 minutes. The mice receiving antivenom mixed with venom survived actually throughout the entire period that they were observed in, but here you can see the first 4 hours. And now to the real exciting stuff. This is how it looked when we pre-incubated a single monoclonal IgG antibody with lethal amounts of Nycalthia whole venom and injected it intravenously in these mice. All mice survived the 24-hour period, which is what is recommended observation period from WHO. But as the mice were not doing super well and some of them were showing signs of neurotoxicity, we actually decided to extend the observation period in one group up to the 72-hour mark. And actually this 72-hour mark, the highest dose of 1:4 toxin to antibody molar ratio there was full neutralization of lethal amounts of Niacalthia whole venom by a single monoclonal antibody in 3 out of 4 mice. So to the extent of our knowledge, this is the first ever monoclonal human IgG to neutralize the lethality of any animal whole venom. And we're quite excited about this. And we think that this kind of gives us the direction that our ambition of replacing traditional antivenoms at some point in the future with these recombinant antivenoms is feasible. And even though this is a very simple venom, we don't believe that we'll be able to use monoclonal antivenoms for all other snakes, or maybe even for this snake in particular, but we believe there is a future for oligoclonal based recombinant antivenoms. And of course, a project like this is not undertaken by any means by myself alone. So I would like to acknowledge all the people that were involved, the people from DTU and the Tropical Pharmacology Lab, especially also Kim from Sophion Bioscience that enabled all the electrophysiological work. To a very high degree, Bruno and Chema from Instituto Economia Picado for doing all the animal experiments already back since 2015. And also in particular, Anish and John and Ayantas in general for allowing me to come there and do a lot of the experiments at their company, but also for providing us with the library that we've used for the selections. And then to all the people that funded the project. Thank you.
Thank you very much, Line, for your presentation.